Okay, sweet. The recording should all be all set. All right, let's go ahead and go through activity one, talking about circulation and gas exchange. First one, what is the function of a circulatory system? Do all organisms need? Who would like to take number one?
I can go.
Thanks.
So basically the function of a circulatory system is to exchange materials with the environment and the organism, so like oxygen and carbon dioxide. And then not all organisms need a circulatory system. For example, like cnidarians and flatworms don't have a circulatory system because they're so thin that they don't need one.
Exactly, yeah. Nice. Number two, what do some organisms have, or why do some organisms have an open or a closed circulatory system? What are the differences? Cole, would you take number two?
Sure. So organisms that have open, they have a different type of fluid, which is known as hemolymph. Hemolymph is just like an interspatial fluid rather than fluid that goes through the veins. And so it's able to just diffuse into the organs that are being soaked by this fluid. Closed circulatory system is more complex. It allows for chemical exchange in the capillaries. It has a higher blood pressure, and larger organisms tend to have this because the blood can travel longer distance.
Yeah, nice. Yeah, and then hemolymph versus blood, that's a big distinction, and then especially that blood pressure too. We'll talk about this a little bit later with the capillaries, but in organisms with a closed circulatory system, it's like a cell, a little bit of fluid outside of it called the interstitial, and then there's the capillary with the blood and stuff in it. So you have the blood and interstitial fluid as separate, whereas with organisms with an open circulatory system, you just have hemolymph, which is like all of that fluid mixed together, and then the cell's just sitting nearby. Nice. Thanks, Cole. Number three, compare and contrast single circulation versus double circulation. Maddie, what'd you have for this one?
I had that the single circulation was less complex than the double circulation, and that it's found in fish. Single is found in fish, and then double is found in amphibians and mammals.
Yeah, exactly. And then for which one of those does blood get oxygenated and deoxygenated in the same circuit?
Single.
Yeah, exactly. Yeah, and that's kind of the big difference with the single is that the blood going in and coming out of it's deoxygenated, and then it goes through the gills, goes through the rest of the body, and then it comes back to the heart. Whereas in double circulation, you basically have one circuit to oxygenate the blood, and with like the pulmonocutaneous one, and then you have another circuit to, the purpose of it is to get blood to the body, but then it ends up getting deoxygenated and then coming back. Sweet, thanks, Maddie. Number four, describe the path of blood flow from the inferior vena cava throughout the entire circulatory system of a person. And then could you also note at which points the blood becomes oxygenated or deoxygenated? Jenna, would you like this one?
Yeah, it's going to be a long explanation, but bear with me.
Take a big breath.
So the inferior and superior vena cavas return deoxygenated blood to the right atrium of the heart, and then from there, deoxygenated blood flows through the tricuspid valve into the right ventricle, and then the right ventricle leads to the pulmonary valve, which leads to the pulmonary trunk, and then that splits into the right and left pulmonary arteries, which take deoxygenated blood to the lungs. And then from that point, blood becomes oxygenated and returns to the left atrium through the pulmonary veins. And then the left atrium turns into the bicuspid valve that leads into the left ventricle. And then the left ventricle goes into the aortic semilunar valve, which leads to the aorta. And then it goes through the ascending aorta, aortic arc, and descending aorta to the body tissues. And then from that point, the body tissues take up the oxygen, and then deoxygenated blood is returned to the heart.
Nice. Thanks, Jenna. That was really well said. A couple fun things, just because there's so much going on with this. Y'all have probably heard that arteries carry oxygenated blood and veins carry deoxygenated blood. Actually, that's very commonly true for just almost everything except for a few things. So make sure to note that, just as Jenna was saying, when you have blood coming into the heart, it's deoxygenated, but then it gets pumped to the lungs. And so that thing is called a pulmonary artery that it's going through. It's a pulmonary because it's going to the lungs, artery because it's leaving the heart, but it's deoxygenated blood inside going to the lungs to get oxygenated. And then once it gets to the lungs, it's in a vessel coming back to the heart, so it has to be in a vein, but that blood is now oxygenated, so you're having oxygenated blood in a vein. Those are the only two vessels that I know of where the typical arteries carry oxygenated blood, veins carry deoxygenated blood is not true. So just keep that one in the back of your mind. And then also, does blood go from arterioles to arteries or from arteries to arterioles?
Arteries to arterioles.
Arteries to arterioles. Yeah, arterioles are like smaller versions of that. And then does blood go from veins to venules or venules to veins?
Venules to veins.
Venules to veins, yeah. So you have artery, and then, so the arteries are kind of big, then to the arterioles, which are smaller, then the capillaries, and then venules, which are like the small veins, and the venules go together into the actual veins. So just make sure you know which, like the flow of blood between those like 5 things. Does that make sense? Okay. Do y'all have any questions on for the pathway or when blood's getting oxygenated or things like that?
Mm-mm.
Okay, cool. Thank you, Jenna. All right, we'll move on to Cinco. What is the difference between systole and diastole? Sophia, what did you have for this one?
So I said that systole was basically the contraction phase of the cardiac cycle, and then diastole is the relaxation phase.
Exactly, yeah. Yeah, that's really all there is to it. Nice. Number 6, how do veins and arteries differ? What chemicals are used to signal for vasodilation and vasoconstriction? Haley, what do you have for this one?
So I said how like for the veins and arteries, veins are usually going to the heart and arteries are normally going from the heart and like carrying oxygenated blood while veins are carrying deoxygenated blood. And for the vasodilation and vasoconstriction, nitric oxide does the vasodilation and then it's peptidal endothelin or something for the
peptide endothelin. Yeah.
Thank you. I can't read my own notes for the constriction.
Nice. Yeah. And this is for everyone. Which one is thicker? Are arteries or veins thicker?
Arteries.
Arteries. And what component of the artery makes them thicker than the veins? They have more what?
Oxygen? Or
She said like they have elastic walls.
Yes. They have elastic walls, but then inside of those, they also have a lot of smooth muscle. So smooth muscles, you typically hear about that in the GI tract. That's what is like peristalsis. It's that involuntary muscle. It doesn't have striations. It's usually not really powerful in terms of contractions, just kind of loose stuff. So it's really common, and there's a lot of it in the arteries because the arteries are at a really high blood pressure. Because the blood getting pumped out is getting just squeezed super hard by your heart. And so if your arteries were really thin, they would just rupture and then you would bleed out internally, which would really not be good. So to counteract that, they have a lot of smooth muscle that contracts really hard, which then squeezes the blood in, which actually increases the blood pressure even more. And that also helps propel the blood all the way throughout your body. If there wasn't any of that contractile force or pressure, your blood would just kind of trickle around your body kind of whenever it feels like it, and you would not be getting enough perfusion. You'd not get enough blood flow to all the parts of your body that need it.
So what do veins have then?
Oh, great question. Yes. So smooth muscle is the way that arteries make sure blood's going forward and not just sitting around or going backwards. How do veins prevent blood from going backwards? What do they have?
Don't they have like valves?
Exactly. Yeah, they have valves. So veins actually only pump's not the right word to use. Blood travels in veins when your other muscles contract. So like if you're just sitting down, that's why like your foot or something might fall asleep because there's nothing getting the blood flow out of it. And, well, there's actually a couple of reasons for that. But if you're just sitting down all day and you're not like moving your legs at all, your blood is just going to pool in your legs because there's nothing naturally to send the blood back. But when you start walking, the contractions in your calves will squeeze your veins, and that pushes the blood up. And then you have valves in your veins to prevent the blood from flowing back. Because otherwise, if there were no valves, your blood would just pool at the lowest part of your body. But those valves make it go up a little bit, and then it reaches another valve, so it stops there, and then it keeps going up and up. Eventually, your blood gets back to your heart. So, neat. Okay, thanks, y'all. Number 7, what happens in the capillaries and what affects this? Viraj, what did you have for 7?
So we said capillaries, their main job is the exchange of substances between blood / and I forget the name. It starts with an I. It's
interstitial fluid.
Interstitial fluid. Is that how you say it? Can you say
interstitial? Yeah. That's a weird
interstitial fluid. Yeah, and then and also I think what she was also saying, they constrict and dilate as well, depending on like how much or like the rate at which materials they need to travel with, or like how much, or maybe I'm wrong on that part, but
no, that's yeah.
Mm-hmm. That's
yeah, so if you think about like capillaries if you're running from a bear, do you need to digest the snack you ate beforehand? Probably not. You probably just want to run. So all of those sphincters leading to your GI tract are going to squeeze, and that way blood can't get into the capillaries in your GI tract, and instead the blood, like it won't be able to enter there, so it'll have to go somewhere else, probably your muscles, so that way you get perfusion there. So there's little like sphincters that constrict at each of the capillaries to prevent blood flow from getting there, so forcing blood other places.
Kind of like not necessarily relevant, but is that why you get cramps when you run?
Oh, like stomach cramps?
Mm-hmm.
That's a good question. I don't know.
It's not that hard. It just sounded right.
I'll look it up when we split over groups and see what the stomach cramps while running. And I'll get back to you. Do y'all have any other questions on, also in 7, blood pressure. Okay, does blood pressure tend to push things out of the blood vessels, like out of the arteries, or does it tend to push things into the arteries? If the pressure is really high inside of a tube, does that tend to make the things inside the tube go outwards, or does it try and get more things into the tube?
Push outwards.
It wants to go outwards, right? Think of like a water balloon when you're squeezing it. Like that water, because you're increasing the pressure, it wants to pop out somewhere. So the same thing, your blood pressure is naturally going to force things out of your vessels. It's going to try and push nutrients and cells, oxygen, all that kind of stuff out of your blood vessels. But if that were the only force working, you would like lose everything. Like everything inside of you would perpetually leak out of all of your arteries, which would not be very good. So instead, there's another pressure that works against it. Do any of y'all know what that pressure is?
Partial pressure?
Osmotic pressure.
Osmotic pressure, yeah. And do you know how the osmotic pressure works, Viraj?
No, I just saw it in the diagram.
That's okay. Yeah. Does anyone know, like, what is the thing that drives osmotic pressure?
It goes from an area where there's like low amount of water to a high amount of water, or
yeah.
Like that.
Yeah. So inside your veins or your arteries, or well, either way, or your capillaries, you have all sorts of proteins that are too big actually to go outside, so they're not forced out with the blood pressure, and they tend to work as sponges absorbing water. And so with those proteins inside your blood, they kind of take up the water that's there, and therefore there's apparently less water. It appears there's less water inside your vein, and therefore it draws more fluids in from outside. So there's proteins that kind of work as a sponge, and that's drawing stuff in, which kind of counteracts the blood pressure pushing stuff out. And that way you don't perpetually lose all of your fluids from your vessels, and nor do you take in too much that would cause them to rupture.
So that just allows like all the nutrients to go out and for you not to like die of lack of water.
Exactly, yeah. The blood pressure is going to work, especially at the start of the capillaries, to push things out. So as you see like blood going through like artery through a capillary then to the vein, as it starts, the blood pressure is much higher than the osmotic pressure. So that forces stuff, especially like oxygen, nutrients, all that kind of stuff out of it. And then as you go further along in the capillary, the blood pressure is weaker, but the osmotic pressure is really strong. And so that tends to suck things back in, particularly fluids or carbon dioxide or other things like that. So that way you're able to give nutrients and then also take back fluid at the end of it. So y'all can worry about the more fun details of that when you get to HPhys, but this will be good enough for now. There's lots of other fun things with it, but again, more shenanigans for later. Okay, let's jump to number 8. Where do stem cells originate and what do they create? Did any of y'all draw out the little chart for this? Okay, Maddie, would you share your chart with us?
Yes. Want me to show the picture like from my screen, or don't want to show like what I drew?
What you drew, if you don't mind.
Show it like this. No, I'm gonna put it backwards. So it originates in the stem cells, or the stem cells are at the top of the diagram, and they're in the bone marrow. And then from that, there's 2 main branches. So one of the branches is the lymphoid progenitor cells, and those are the B cells and the T cells, which are lymphocytes. And then the other branch is the myeloid progenitor cells, and those are the erythrocytes, the neutrophils, the basophils, monocytes, platelets, and eosinophils.
The eosinophils. Yeah, lots of fun words with all the immunology. Nice, thank you, Maddie. Yeah, so that's kind of the map of how all of the cells in your blood start off as stem cells, and then later they kind of can go down these different pathways. And there's even a larger chart with, I think, like 30 or 40 if you really want to get wild and see all the possible derivations of it. But save that for immunology or med school or PA school or whatever. This will definitely work for now. One thing to look at, just the names of a couple of these things. Do any of y'all know what the suffix CYTE means? Cell, exactly. So all of these are different ways of saying like monocyte is a singular cell, which monocytes kind of have their own function later. But erythrocytes, erythro like red cytes cell, so like red cell, or lymphocytes, lymph cells. But yeah, I would just know this chart. It's kind of a handy thing for remembering how you get stuff. And especially when we get into immunology, this actually is a little bit helpful to kind of see what gets grouped with what, especially when you get to B cells and T cells. But that'll be fun for Thursday.
What about phils?
About what?
Phils, neutrophils, basophils.
Oh, neutrophils, basophils, and eosinophils. Mm-hmm. Those are all specifically for your immune system. Those are also cells. I'm not sure what the suffix "phil" means, but I can look that up. But eosinophils they fight parasites, so do basophils, and then neutrophils do something a little bit different. But I think we'll get to that at the end of activity 2. So we'll go into a bit more then. But thank you for asking. Okay, let's see. We only have, oh boy, we still have 3 or 4 more. So we'll try and race through these. Why are respiratory surfaces wet? Will, what'd you have for this one?
I kind of struggle with this one. I guess I said it was wet to like help. I said it was like, I was so confused. I said it was wet because it would help the, I guess, the exchange of, it would help with the exchange of nutrient or the exchange of gases. I'm not entirely sure.
No, that's exactly it. It seems a little bit counterintuitive that gases would be better exchanged through a liquid than just through like open air. But for whatever reason, that's just how it works. And so by having, especially if you think of like the alveoli in your lungs, they have a huge surface area to volume ratio by being these clusters like of a bunch of little grapes. So there's like lots of surface area all over it. And then by having a super thin layer of fluid, it's really easy for gases to diffuse from the blood to the fluid to your lungs and vice versa. So a little bit odd, but it just, that's what allows for the best gas diffusion. Nice one. Number 10, how do aquatic animals maximize their oxygen uptake? Jordan, what did you have for 10?
Sorry, I was still writing down what you said for 9. Lost my train of thought. So I thought, I could be wrong because I got a little confused on 10, but I said that it had a countercurrent exchange system that allowed it to take up roughly over 80% of the oxygen dissolved in the water.
Yeah, nice. And how is it countercurrent? Like what's flowing in what direction that allows this to work well?
I don't know.
Like if the fish is swimming to the right, let's say the fish is swimming like this way, which way is it going to pump the blood? Is it going to pump the blood also to the right, or is it going to pump it to the left?
To the left.
Okay, this is kind of the weird part of the countercurrent. It's swimming forward, and so effectively the water it's encountering is coming at it this way. And so as water comes against it this way, it's going to pump blood at that same direction. So that way there's lots of, like if fish is going this way and it's pumping blood this way, effectively the blood is moving really fast in this direction. So it's encountering a lot of water as it's going across. Whereas if it was pumping water like backwards, then as it like moved along through the ocean and pumped blood this way, the blood would be interacting with effectively the same part of the water the entire time. And so there'd be less interaction. And so that pumping of blood forward as water is going backwards is countercurrent, like we talked about for, was it an SI or two ago, for maintaining temperature, that by having those kind of cross directions, you get a lot of exchange going on there. And that's what leads to that massive uptake of oxygen, which, yeah, it just seems really high, but it's just that efficient. And then also fish can, they can swim forwards, or they can also, y'all probably have seen this where they move their little fins or flippers and they basically are pumping water against their gills. So that way, even if they're not moving forward, there's still a circulation of fluid that allows them to uptake more oxygen. And one other tiny little thing there, sometimes people talk about a partial pressure gradient going on with the oxygen. Do any of y'all know what that means?
It means that the oxygen is going to go from the area of higher, it's going to diffuse from the area of higher partial pressure to the area of lower partial pressure.
Exactly, yeah. And comparatively in this example, there's more oxygen in the water. And since there's less oxygen in the fish's blood, it's going to move from the water into the blood. Nice. Okay, jumping on to 11. If organisms don't deliver O2 in their blood or hemolymph, what kind of system can they use? Who had 11? Lauren, did you get to 11?
No, I did not. I just finished number 10, actually.
Oh, so close. I think one of y'all got to. Haley,
I can try. So it uses the tracheal system, and basically it's just a giant tube that's constantly open, and it's meant more for animals like smaller, like insects or those that are flying due to like consumption rate. So that way they have constant access to air always being in there, and then they can, like, even if they can't deliver it to their bloodstream, it just comes in naturally and then just goes out naturally.
Yeah, exactly. And it's especially common too in smaller organisms. Imagine if we tried to have little trachea going to every cell in our body, trachea are not that strong, so just the existence of like the weight of our head and shoulders pushing down would crush the itty bitty trachea all over our chest. So it would not work well in large organisms. Instead, we just put our gas in our blood and use the circulatory system. But certain ones will just use the trachea. Nice, thanks, Haley. And then, all right, finally the last one. How do partial pressures cause O2 acquisition in mammalian lungs? We talked about this a little bit, but how in particular is this working with O2 and CO2 in mammalian lungs?
I can try this one.
Yeah.
I was like just trying to figure it out. But like we said, the partial pressure is the pressure exerted by a particular gas and a mixture of gases. And so basically in mammalian lungs, where like gas exchange happens in the alveoli, the O2 that we need for our cells is going to be moving in because we're at a lower concentration, and the CO2 is going to be moving out because that's what we're trying to get rid of. So basically that's going to help us get more O2 into the alveoli for our cells to use.
Nice. Yeah, well said. Okay, sweet. Do y'all have any questions over activity 1? No? Okay, let's go ahead and jump to activity 2. There are, I think there's only 7 questions, but they're all a little bit larger. Don't get too worried about 11. I can just explain, actually, I'll just explain 11 now because it'll make it a bit easier as we're going through it. But when we go through the immunology, all the immunology things, they use a couple terms and they have a little bit slightly different meanings. So there's the word pathogen is this just general term that means anything infecting the host organism. So a pathogen could be a virus, it could be a bacteria, it could be a fungi, it could be any of those things. It's just kind of a generic thing to say the invader that your immune system is fighting. Peptides are just chains of 2 or more amino acids, so just think little like protein-like chains, or one is like a protein. Antigens are particular things on the outside of a pathogen that the immune system targets. So that would be like if this is your cell and there's something like hanging off the outside, that's something that our immune system could use to recognize this cell as an invader. So that thing would be called the antigen. And then the particular little spot on that antigen where our antibody or whatever would bind to, that's the epitope. So epitopes are little things on the antigens. Antigens are usually peptides, but they could also be polysaccharides or a few other things. And then that all happens on a pathogen. Okay, so that's 11. Hopefully that'll help you with the others. We're gonna go ahead and split back into breakout rooms, and then we'll come back in a little while, maybe 7:45, and talk through everything. So see y'all in a few. Okay, recording is up, so let's go ahead and get rock and rolling over activity 2. Number 10, what is the immune system and what do its main branches do? Who had number 10?
I had number 10.
Sweet.
So I said the immune system was to protect against invasion and pathogens. And then the 2 main branches are the innate immune system, which is found in all organisms, and the adaptive, which is only found in invertebrates.
Awesome, yeah. And what, generally speaking, does the innate do? Is it like fast or slow, specific, general?
Isn't it general and slower?
The innate one is general, but the generality allows it to work faster. So like something the innate system would do is just kind of have like an acidic environment on your skin. It's just kind of a general thing that's all about. It's just going to kind of kill any sort of thing that might be on it, but therefore it's fast. Whereas in the adaptive immune system, this would include things like antibodies that take a lot of time to form because they have to get a really precise basically binding interaction with the different peptides in the bacteria. And so sometimes that takes a couple days or even like up to a week to do. So it takes longer to do it, but it's very specific and effective. And we talked about this in a couple of the different breakout groups. The innate immunity, you find it in invertebrates because most of the invertebrates are only going to live sometimes a handful of days or weeks. So if they got infected with a certain disease and survive, they really don't need to have the kind of immunological memory to fight off that same disease, because they're probably just not going to get it twice. Or if it kills them, it's okay because there's a million more bees in the hive. Well, actually, we need more bees, but other insects. Whereas for us, well, if you want to live your full like 80 years of life, you're probably going to catch some of the same things like strep or other diseases multiple times, and so you want to be able to develop a stronger response to it afterwards. And just kind of going along with the general process of us being more complex organisms than insects, it kind of makes sense to have a more complex immune system. But yeah, we can get into that a bit later. 11, we already kind of went over. Hopefully y'all saw that come up, and it'll come up again, especially epitopes and antigens when we talk about the adaptive immune system on Thursday, probably. Number 12, how does the innate immunity of invertebrates function? Who had 12?
I did. So in invertebrates, they have an exoskeleton and a chitin-based barrier and lysosomes. And they have hemocytes, which do phagocytosis and have antimicrobial peptides. And with that, they can recognize structures on the pathogen cell walls.
Nice. Yeah, and exactly that, generally working by recognizing patterns on pathogen cell walls. So this is, well said, Maddie, this is a big difference between the innate and the adaptive. In the innate, they're just going to recognize like, oh, this is a peptide that's often found on bacteria. So if I find this peptide on a certain cell, it's probably a bacteria cell, we should probably just destroy it. Whereas the adaptive immune system would be like, let's get a very specific response to one exact protein found on one kind of bacteria, and then only fight that one. But since we're talking about innate, it's much more general. Nice. Okay, number 13. How does the innate immunity of vertebrates function? Who had 13?
I did. Okay, so in vertebrates, they rely on toll-like receptors, which are basically recognition proteins that produce signals to initiate responses tuned to the invading microorganisms. And these responses can include either killer cells, interferons, and inflammatory responses.
Exactly, nice. Yeah, the inflammatory response, think about, that's definitely something very specific to more complicated organisms like vertebrates, because if you're an insect, there's not really any room to have inflammation, nor do they have a closed circulatory system to even send more blood to that area. Yeah, and the toll-like receptors are just like one really good example of something that is a particular mechanism that we have that recognizes like flagella or DNA or lipopolysaccharides of bacteria. It's still general, but it's able to recognize those patterns on multiple different pathogens and then knock them out in some way, shape, or form. Sweet. Thanks, Joy. Number 14. How do some invertebrates protect themselves against viruses? Who had 14?
I had 14. So the way it works, it's also like another recognition type mechanism. So the thing about viruses is that their genetic material is RNA, and when they replicate their RNA, it first is a single-stranded RNA, and then it becomes a double-stranded RNA when it replicates, and then it becomes a single-stranded again. So the way it works is that the enzyme dicer2 recognizes the double-stranded RNA during its replication, and that enzyme cuts the double-stranded RNA into fragments, and then a protein complex that contains the enzyme Argo binds onto one of those fragments, and then basically ejects one of the strands, so it only has one strand in that protein complex, and so then it travels around binding to complementary strands, and when it binds to those complementary strands, it breaks it apart and basically kills the virus.
Nice. Yeah, exactly. It's recognized that double-stranded RNA, and then it's breaking that up, using a part of that, and then it uses that to recognize the same virus later on. Nice. Okay, number 15. How do Oh, okay. Yeah, go ahead.
Sorry.
You're good.
So neutrophils, basically they explode and release these traps that trap the pathogens. Therefore, that's what it means by like they sacrifice themselves. And then like being able to trap those pathogens, that's basically how they kill them off, or they do like phagocytosis. And then macrophages are in bone marrow. They can do phagocytosis, and they also have antigen presenting cells. But what's important about macrophages is that they secrete signals to help activate other cells to help fight off infections. And then natural killer cells just detect abnormal cells and then release chemicals for cell death.
Nice. Yeah, all sorts of things going on with these, and we'll see even more cell types. So as we're getting through all of these, I don't know if you'll like flashcards or other things, but try and find ways to distinguish them, because by the end of this, there will probably be like 7 or 8 different types of cells. And it would be very easy for Dr. Fernandez to ask you like, oh, which of these 8 things does this one particular function, or does these 2, but not this other thing? Which one does phagocytosis, but is also from the bone marrow? So I would make sure to know all of these and their kind of classifications. Well, this is a little bit like going to all the classification stuff from last test or 2 tests ago. So yay for that. But yes, neutrophils and macrophages are the 2 doing phagocytosis here. Macrophages are really important. We were talking about this in one of the groups. The immunological system does a lot of communication, like an absurd amount. And the macrophages are kind of one of those key components. And so by phagocytosing a bacteria, they're killing that one, but then they will also break it up and then take little parts of the antigens, the little proteins from that, and it will present it to other cells and say, hey, this is the bacteria that I just gulped up and destroyed. Go make an even better response to this one. And it presents the antigen to that other cell, and then the other cell runs off and activates the adaptive system. So that's what it means by the macrophage. She says professional antigen presenting cell. The reason it's professional is because there's like more casual antigen presenting cells too, but that's a distinction y'all don't need to worry about for this class. But yeah, and then natural killer cells, this is the one that's not doing phagocytosis and instead of just dumping chemicals on it. And sometimes you'll just see that written as NK cells. That's just natural killer. So neat. Thank you, Sydney. And now we're on to the last one. So we might actually finish on time. What do you know? Okay, 16. What do eosinophils and dendritic cells do, and how are they similar or different from the cells that we just talked about? Who had 16?
I did number 16. So the dendritic cells stimulate adaptive immunity against pathogens. And then the eosinophils defend against multicellular invaders. And when they do that, they also discharge destructive enzymes.
Nice. Yeah, and here again, we see some of them are having enzymes, and some of them, like the dendritic cells, do phagocytosis. Dendritic cells are another antigen-presenting one. So they're a little bit like the macrophages, where they're going to phagocytose it or phagocytose something and then present the antigens to someone else. One particular things with the dendritic cells is that they're typically found near the epithelial cells. So they're more like the outside of the body, whereas macrophages are more likely to be found in all parts of your body. And then eosinophils, yes, they're particularly fighting parasites. There's also basophils and some other ones, but we will leave that to Thursday. And then the eosinophils just discharge enzymes. Whew, okay, that is a whole lot, and that was only, I think, the first like 10 slides of the immunology PowerPoint. So we will most likely spend a lot of Thursday doing that, and probably some review time. I'm leaning towards just having Thursday as like mostly immunology, and then sometime in the dead days doing review stuff. What would y'all prefer though? Would you prefer Thursday to be more immunology or more like a big review session for everything?
More immunology.
Immunology. Okay, sweet. We'll plan for doing immunology on Thursday, the rest of the PowerPoint, and then Katie and I will have a big review PowerPoint, a practice test, which I think we got up to like 140 questions from last semester, because I was studying for the MCAT one night and I just got tired of it, so I just made a bunch of questions. So we'll probably have to whittle it down so that way we're only giving y'all questions that are relevant to the things that you test on, unless you just want a massive amount of questions about everything 1306, but you probably just want the ones that are actually helpful. So we'll hopefully get that out to y'all sometime this weekend, and then maybe Monday or Tuesday we can do something that's more like extended office hours or something like that to go over more final review things. Does that sound good?
Yeah, and then I'm also open to maybe doing like review over the weekend even. I don't know if that would work.
Oh yeah, would y'all prefer doing something on the weekend or during the dead days?
Either.
Whatever's easiest for you.
Okay. I'm inclined towards the dead days just in case people are doing things with their family on the weekends. I don't know how much y'all's schedule is still like a Monday to Friday work day at all, but hopefully Monday, Tuesday, everything will be a bit more lax. But I'll see if a bunch of y'all really want on the weekend instead. We can do that too.
I think the test is Wednesday.
Oh, that's right. Okay, yeah, some of y'all have Wednesday instead of Friday. Okay, yeah, then we can shoot for either Saturday or maybe early on Monday or something like that to give y'all more time. Okay, sweet. All right, well, feel free to hang around if you have more questions. If not, have a good night. Have fun studying. And I'll see y'all Thursday.
All right, thanks.
Thank you.
See you guys.
Thanks, Liam.
Thanks, Liam.
Thank you.